Good morning. Cellular agriculture is a vastly growing field of study and needed research focused on the optimization and control of culturing muscle cells into commercial products. At HMT, we specialize in measuring and understanding cellular metabolism with a new focus on muscle cells for cellular agriculture. Understanding cellular metabolism is more than just measuring metabolites. It is understanding their pathways and regulation. In cell culture, regulation is key. Corey said it in 1929 that understanding metabolism means understanding circular feedback and control, how metabolites interact with each other in their reactive products. While Corey and Gertie Corey received their shared Nobel Prize in 1947, four other couples have also received Nobel Prizes. The Curies, both Pierre and Marie, as well as their daughter and her husband 30 years later. Gunnar and Avel Myrtle and Edward and May Moser all received their Nobel Prizes as couples. What are the critical pathways or metabolism to understand culturing muscle cells from chicken or fish? How different is the biochemistry between different species? Where do we study, media, cells, or both? What are the essential nutrients in fetal bovine serum, and how are essential nutrients different from different cultures? Applied metabolomic studies to cell cultures is not a new application, however. Such applications are increasing at a significant rate due to improvements in growth in metabolomic technologies in parallel with growth in demands of cellular agriculture. As an example, culturing fish cells is finding to be very demanding, requiring fresh discovery and pathway construction. Essential nutrients can vary significantly between different species. Understanding chemical processes for biomass and energy generation will take a comprehensive study in creation of new pathway maps. Cell proliferation is one goal for successful culture. We typically normalize cell metabolites with cell count or cell mass. Does metabolite concentration relate to cellular proliferation? While increasing yields of glutamic acid have been suggested to be an indicator of cell growth or proliferation, is it uncertain that this is so? Cell growth is an essential requirement in interest in growing muscle cells to product. To promote growth, we need to understand what are the essential nutrients, what may be hindering growth, and how these factors change over cultivation period and conditions. Lastly, how close are these requirements in culture to in vivo? To start, what are the main pathways that we must understand for cell growth? Biomass generation, energy production and generation, response to oxidative stress, and detoxification of high levels of free radicals, lactic acid, ammonia are among the primary areas to understand. Within these pathways, there are a number of metabolites of different classes: zooionic amino acids, neutral sugars, phosphorylated nucleic acids, waste products like ammonia, urea, lactic acid, free radicals, hydrogen sulfide, water-insoluble fats and lipids, and cofactors such as vitamins and minerals. A metabolic study then requires more than just one assay to capture this diversity. Understanding how these diverse molecules interact leads us to pathways and regulation and understanding cell culture optimization. Measuring metabolites in media and cell pellets starts the study, then introducing changes in media and composition and cultivator conditions with the design to align or discover which pathways or metabolites are driving or hindering cell growth can create unique analysis from different species. Time course studies are normal requirements, changes over time linked to cell counts or cell density with changes in media and nutrient composition. Alternatively, we may examine metabolites or changes replacing animal-based media. Once we collect data, we can then organize the metabolic readout by assigning metabolites to one or more primary regulated biochemical pathways. These pathways, or circles, consist of rate-limiting steps, specific gene activation, reactants and products, and factors that can both activate or inhibit the rate-limiting steps. While there are a number of different pathways of interest, we often start with energy metabolism and glycolysis. These pathways are essential for cell proliferation and biomass generation. They also generate ample amounts of radical oxygen species and ammonia that can act as negative regulators. One hinge point for energy metabolism is the outcome from the production of pyruvic acid. Pyruvate can be converted to the endpoint lactic acid, react with ammonia to form alanine, or transport to the TCA cycle for fatty acid synthesis and ATP generation. How the cells distribute pyruvate is essential to understanding cell growth. Here are three examples of measuring lactate, pyruvate, and alanine in 3-cell media under different culture conditions. In condition A, lactate increases over time, while pyruvate decreases and alanine remains stable. This is a common observation in healthy cells. In condition B, while lactic acid increases, pyruvate decreases but at lower levels than in condition A, and alanine shows a slow increase. Does this suggest cells with a higher level of ammonia or an increase in BCAA metabolism? In condition C, lactic acid and pyruvate increase and decrease at a much steeper gradient, with much higher levels of lactate and lower levels of pyruvate, with high levels of alanine. What does this suggest? Perhaps higher proliferation with nutrient depletion. At HMT, we use our own pathway maps assembled from our observational studies from 20 years of operation and using our extensive metabolite coverage. Let's examine the glycolytic pathway and focus on the fate of pyruvate. In this blow-up, we see that the pathways and options for pyruvate, the enzymes involved in these pathways, and examine the factors that influence these enzymes to help understand control of this pathway. Another first focus is the distribution of glutamate and glutamine. In the same three cultures, we see three different patterns of glutamate and glutamine. In A, glutamine declines while glutamate increases over time, seemingly coupled together. In B, they are still coupled, but both increasing with higher levels of glutamate. What does generate glutamate, and why is glutamine acting in the opposite direction of culture A? Why in condition C, glutamate and glutamine seem to be uncoupled, acting independent of each other? What does our pathway map say about glutamine and glutamate metabolism? We may start with the urea cycle, which maintains the redistribution of ammonia following deamination of BCA amino acids, an important process in muscle metabolism. 2-oxyglutarate can pick up and release ammonia for the biosynthesis of glutamic acid. The outcome of newly formed glutamate, 2-glutamine, or other cyclic outcomes can be regulated by alanine concentration, ammonia levels, or BCAA flux, as well as downstream outcomes. For example, GABA is a major regulator of protein synthesis. Glutamine is a major precursor of GABA in muscle cells. I've mentioned ammonia a few times. The principal pathway for handling ammonia levels and regulating nitrogen metabolism is the urea pathway, regulated by N-acetylglutamate. However, the urea cycle in muscle in vivo may not be regulated in the same way as in vitro. In vivo, alanine becomes a waste product along with lactic acid, both of which can be transformed by liver and recycled back as glucose for muscle metabolism. However, how does this work in cultivated cells? In this example, 4-guanidinobutanol is observed mostly in condition B, increasing with time. How does this not so common metabolite factor into muscle metabolism? 4-guanidinobutanol, in fact, is found in the urea cycle as an alternative pathway to GABA. So let's look at the urea pathway and focus on GABA synthesis. Now, focus on this pathway, we see that 4-guanidinobutanol is on the oxidative pathway to GABA versus a non-oxidative pathway through polyamine synthesis. As GABA is a major requirement for protein synthesis, the pathway to synthesis can be influenced by oxidative stress and peroxide clearance. Polyamines are also known to be essential for cell growth. In the same three conditions, the highest levels of polyamines in media are in condition C. What is influencing these high levels? While polyamines are required for growth, high levels may be cytotoxic. How are these higher levels connected to the steep changes in lactic acid and pyruvate as we see in C? Again, we turn to the urea cycle. The primary drive for polyamine synthesis is the conversion of arginine to ornithine and to ornithine to putrescine. The drivers for this conversion include downstream outcomes like spermine and GABA, as well as levels of vitamin B3. Let's look at a few nucleic acids. In this same C culture, there are also high levels of cytidine and guanosine. Why only these two nucleic acids? What is unique with these? Well, these bases are natural chelators, meaning they form self-assemblies under proper concentrations and conditions. Is the ion pairing capabilities of the polyamines we just saw in C related to the high levels of these nucleic acids? Are these nucleic acids forming a scaffold, and how does these relate to the high dynamic range of lactic acid and pyruvate? We can't avoid looking at lipids as well, as certain fats are essential for cell proliferation and growth. In conditions in A and B, these essential fats decrease over time, while condition C shows a dramatic loss consistent with rapid release of lactic acid. High proliferation is depleting these essential fatty acids from the media, perhaps driving fatty acid synthesis at the expense of protein synthesis. What lipids are essential? It may depend on the species of muscle, not just cultivated conditions. Fish, chicken, beef are all different. In muscle cells, fatty acid synthesis competes with protein synthesis, hence the proper intake of essential fatty acids is critical to cell growth and density. Handling all this is a network of antioxidants. In condition C, we saw rapid depletion of lipids and rapid increase of lactic acid. We also see here a rapid increase in glutathione as a major detoxifiant for radical peroxides. 2-hydroxybutyrate is a biomarker for glutathione synthesis. Looking at the glutathione-methionine cysteine network, we can focus on 2-hydroxybutyrate as a biomarker for cysteine synthesis. This suggests higher proliferation is required for higher levels of glutathione. Organic acids can also give us insights in cellular metabolism. Ketoleucine is a deamination product from leucine metabolism, driving to synthesis of acetylcoenzyme A in the mitochondria for energy production. In condition C, we suspect that nutrient depletion of fatty acids used for fatty acid oxidation and energy production are being supplemented by BCA metabolism for energy production. In this way, BCA metabolism and glucose metabolism can compete for energy generation. Hence, the metabolism of leucine as a ketogenic amino acid to provide additional acetylcoenzyme A. In addition, BCAAs can activate mTOR and stimulate protein synthesis. So after we examine hundreds of metabolites and dozens of pathways, where are we? The interpretation of pathway activation is an accumulative process of plotting metabolic changes across multiple pathways and providing guidance as the driving factors for biomass generation, energy generation, and detoxification. Adding both cellular metabolites and media composition can also add to these interpretations. In the end, time-based studies, comprehensive metabolite measurements, together with pathway analysis, can lead to optimization of the production process. In addition to cellular metabolism, a metabolic profile of muscle cells can provide data on taste profiles of individual cell products. Our taste library consists of various classes of metabolites like D and L amino acids, nucleic acids, dipeptides, vitamins that interact with taste receptors. As an example, here are pie charts showing different distribution of umami, sour, sweet, fatty, and bitter with changes in cell culture. While not directly telling a product taste, one can see how changes in cultivation affect the distribution of major metabolites that can reflect taste attributes. Metabolomes can also measure essential fats, amino acids, antioxidants, and product development, and how these levels match those of in vivo muscle cells or the natural product. We can quantitate vitamins, both water-soluble and fat-soluble, over different production runs, as well as quantitate amino acids across production lots. To conduct all these studies, we need small amounts of media and small amounts of cell pellets, conducive to cell development. From these small amounts, we can measure cellular metabolites, taste attributes, and levels of amino acids, fatty acids, and vitamins, as well as provide pathway analysis. You can contact us directly or visit our website to find out more and set up a project plan. Thank you.
All right. Well, I hope people can reach out to Alex and his team if they want to get in touch and learn more. So thank you, Alex, for that presentation. Now we can turn it over to Mila, who will present from 908 Devices.
That was a very interesting talk from Alex, where you got a deep dive into the metabolism. And my approach to the topic of analyzing is focusing mainly on the spent media and for cultivated meat. And I'm focusing on capillary electrophoresis, mass spectrometry device as well, but taking the approach of looking into how to gain the information on amino acids in the fresh and spent media very easily. So not going into a full-blown mapping, but of course, or modeling experiment, but maybe gaining the information that can be used for that sort of building the information. So I am from 908 Devices. My name is Milla Nefling, and I'm the market segment manager for the bioprocessing part of the business. we have the pleasure of having a project together with David Block's team at the UC Davis. And I would like to highlight this great article written by Ted in David Block's team. This really kind of sets the scene very well on why media matters in cultivated meat. And so there is so much information about cell culture media, how it should be, what are the best considerations to take. But a lot of that information is for CHO cells. And when we are talking about muscle tissue, fat tissue, and connective tissue, all may be produced and manufactured together in a bioprocess. That is a mighty, mighty good challenge for all of you guys at the other end. And there's a lot of cell culture medias available. There is so much to choose from, but unfortunately, those cell culture medias have not been designed with the requirements of cultivated meat in mind. And so I would like to highlight this article to you. You will get these slides afterwards, and there's a link to this article as well here, but great talk. I have to do a disclaimer though, none of the following slides are from David Block's team. These are from our production, so if there are any mistakes, they are solely ours to take responsibility for. So indeed, there are multiple cells are needed. You guys know this very well. There's muscles, there's supporting cells, there's endothelial cells, adipose cells, and all of the things that Alex just mentioned, knowing what the cells need. We have a lot of that information, but for CHO, and here all these cells, they need to live together, and they need to be growing well and thereafter differentiating. There's a lot of experiments needed to find that great balance of which nutrients these cells actually need in the cell culture media. A lot of information is around, but the challenge is still out there for sure for you guys. So cell culture media, I definitely don't need to tell you this, but the components need to be safe for human consumption and need to be animal component free, antibiotic free, and ideally chemically defined. And this second point is especially important as so many companies, they have to start with a situation where they need to first develop something that is serum free, as that is a requirement. And then the contaminations, they cannot be taken care of by antibiotics. There's a lot of challenges just in that point too. There should be synthetic or engineered growth factors. Growth factors are really important, but again, they should preferably come from a non-animal origin. And of course, these cell lines preferably to be co-cultured so that the end product would resemble the meat product itself. And then very importantly, and if point 2 was a mighty important point, the last point maybe even more so, extremely low cost, so that to give the production the high volume and yet be feasible for actually to be eaten and not just looked at and cherished. So in this picture here, there are a couple of things shown, and the media recycling is something that is highlighted in some articles. And I have to go back to my disclaimer. David Block's team is not in favor of media recycling, but some other people are. And that is then whether you're doing fed batch, which for these long-term cultures that we're talking about for cultivated meat might not be the best option, but then looking at perfusion, looking at continuous processing, the Rebel that I will be showing to you guys and talking to you about, that supports media optimization and quality control wherever you are in this process. So it's not picky to where you where you are. Okay, so if we start with the end in mind, we want to have low cost, great quality product that has the right olfactory and taste taste profiles, but also a spent media that has basically nothing left in it. So because we aim at this low cost, we want to make sure that we only put in our media the things that are needed, the things that are consumed, and at the end, we are not wasting anything. And to get to that, Alex was highlighting in the previous presentation the intricate maps and just all the different pathways that cells do. Usually when we're talking about cell culture, people are looking at physicochemical parameters such as oxygen, temperature, pH, etc. They're looking at nutrients and metabolites when it comes to glucose, glutamate, etc. Looking at cell growth and viability. And of course, in your case, the yield in the final product is the cells themselves. And these different parameters, people are able to gather at line at their bioprocessor. But now, one thing that is often left out is the amino acids. So amino acid analysis is often something that people need to send their cell culture media samples to somewhere else to find out what actually has happened, what has been consumed, although the glucose and lactate and ammonia is readily available information. So now to build a full picture, to get to the knowledge, to get the data input, our approach, what we want to promote is to get all of these different pieces of information at line, including the amino acids. And of course, that leaves a huge number of metabolites untouched, but the amino acids being in such a crucial part in cell growth and cell differentiation, we believe gives a good stab at actually knowing what is happening and being able to control the process. So therefore, I would like to introduce to you the Rebel. This is our 908 devices system, and I can tell you a little bit more about what's inside at the end if I have time. But let's look at what it does. It is a at-line, very importantly, at-line cell culture media analyzer. The sample requirement per analysis is less than 30 microliters. This depends a little bit on the concentration that you would have. We typically say around 10 microliters, which you then dilute and put into the sample vial, and that's it. We cater for all amino acids, and we get the range of analytes every time. There are also a couple of vitamins and biogenic amines, but these are quite often in cultivated meat, spent media so low that this is not really relevant in this particular conversation here today, unless we are talking about looking at hydrolysates, which I will mention in a moment. So it is faster. So you have this at-line analyzer at the bioprocess. So whether you're doing experiments with well plates at this moment of time, whether you're in shake flask type of volumes, or whether you are in bioreactors, the Rebel is so small that it can be right there with you. You take the sample, you spin it down, and then you put it into a sample vial and put it into the Rebel itself. And so this is really meant for you to have that amino acid information right at the bioreactor, whichever scale you are at. It's easy to operate. There's no compromise on data quality based on if you are a mass spec geek or if you are a bioprocess or cultivated meat scientist. And it is a efficient and cost-effective way to just be able to follow, monitor, and develop understanding of your bioprocess more easily. So because they're at line, you can get this information right away. So these results are actionable. Your bioprocess is on the lengthier side, days. So you can see if something is depleting, you can still find that actionable and add something into your feed, into your culture as it gets depleted, and get information on that as well. Right, so this slide becomes a little bit redundant after Alex's presentation, but when we talk about the extracellular environment, which is critical to life, we have amino acids, vitamins, there's also trace elements, and there's proteins, peptides, growth factors, carbohydrates, there's other things that matter. And when it comes to the rebel, that is the discussion point for me today, we are focusing on the amino acids and the vitamins, but indeed with the disclaimer here that the vitamins typically are a bit low in the culture media that you would be using. Okay, so what typically people do today to look at the amino acid concentrations in their culture media, that would be HPLC or HPLCMS. And so you would pull the sample, filter the cells, label and secure collect, and freeze the samples and send them to an expert who can then analyze them and take great care. Now, I'm a analytical biochemist myself. I looked at this 3 to 6 weeks for sample delivery, and I was like, is that really true? But this is actually from our customers. So the real life expectation of when sending samples out to be analyzed somewhere, you do get amino acids. You might get a panel of other analytes as well, but it really does take a lot of time. And we are not as comprehensive as HPLC-MS or the presentation that Alex gave, that human metabolome can give. We are really focusing on these amino acids, but the pain point that we want to address with our system is that you get actionable results at the point of need. So at the bioprocessor, you pull the samples, you spin out the cells, and you dilute in a specific way that's important. You put the samples into the rebel, and you name the samples, and then the rebel analyzes the samples. And this takes 15 minutes per sample, approximately. There's a couple of things, how many repeats, how many samples, blah, blah, blah. But in all cases, 15 minutes, we typically say 10 minutes per sample. Right, so as I mentioned in that next to the burger there, the different types of analytes, parameters that people are looking at, these are now available at the point of need: cell viability, cell density, the ammonia lactate, glucose maltose, etc., and of course the usual parameters. But the amino acids and vitamins, those are typically outsourced, and we now suggest to bring this analysis to AdLine, where you can then look at the results right away. When we are talking to analytical people, they of course have a concern. That box is really small. Is it really going to give me the accuracy of information? It isn't the top notch when it comes to. It is just good. Well, let's say good enough and a bit better than good enough to give you the answers that you need at line. So we are within 20% of expected values, as shown here in a spiked study, and the RSDs, so precision, is within 16%. We of course want to always say 15%, but here you see there is methionine that it has fallen outside that in this particular study. So we have precision and accuracy that addresses the need of knowing what is happening in the cell culture media, but it isn't going there to your 5% mark. That would be a too high of an expectation for this thing here, but it truly works. And the beauty of it as well is that you might have noticed that you only need to filter or spin down the samples. That's because in our system, the presence of protein, let's say that you are doing experiments where you have different conditions that you want to test and reducing the amount of serum. So the serum content really doesn't hinder the analysis. Here, please note that the final reading here is serum on its own, and these different bars here represent the different amino acid readings in the presence of serum in RPMI. And so in these different cases, we can see that we have very tight, the serum content goes down a little bit, but the error bars stay, and we are able to read out the concentrations in the presence of serum without a problem. And you don't need to do any extra protein removal per se. We have here a yeast extract. I mentioned that in some cases the vitamins are attainable when you're doing fresh media analysis to see that your starting point for the experiment is the same. With hydrolysates, you can of course see a lot of variability, as we can see here for different vendors. And here the vitamins are often at the level where we can measure them with the Rebel. So when you want to address and see that, hey, I got a new lot of this extract that I want to test for my cell culture for cultivated meat, what is the starting point? We can see that there's a lot of variability, and it would be smart indeed to check your cell culture media when it comes to amino acid content, because that is really important, of course, in cultivated meat production. When it comes to the hydrolysates, we see a lot of differences. And for example, I've just pulled out 3 different amino acids here: arginine, isoleucine, and serine. And you can see that between the vendors, a yeast extract has a lot of variability, and we can measure this variability with the Rebel. And so you know already a little bit where you're going into when you dissolve your hydrolysate and you put it into your bioreactor experiment. You know a little bit better if you should be expecting the same results as the last time, or if you are somewhere else. So I've taken almost all of the time, but just quickly, the Rebel is indeed small. These are in inches. I don't do inches, I do centimeters, so I have no clue what this is, but I can tell you it's small. It's only 36 kilos, and this is a capillary electrophoresis mass spectrometer, but you don't need a roughing pump, a vacuum pump. Those this is our proprietary technology that we have that makes that the system is small. To operate the system, we provide these kits that have the standards. So it is a mass spec, and you might ask, well, can't I just analyze everything? Sure thing, but these kits are optimized so that we have the standards for amino acids and those vitamins, and so that you can just simply use the standards to do a calibration curve for your analytes, amino acids, vitamins. We have the background electrolyte that is used for running the system, and diluent. Now, this is very important, that the diluent needs to be the one that is provided, and that is then used in the sample preparation. Indeed, just take your sample, dilute it, filter it, or centrifuge, and you're good to go. But it's important to use this correct diluent. And we have this chip. The chip is really important. That is the basis for the separation, and that caters for 200 runs per sample. Now, I will go this through very quickly. If there are any questions, I'm really happy to go through this a little bit more in detail. But indeed, inside the Rebel, what's in the inside the Rebel, there's the autosampler. So you put in your vial. The sample is taken into capillary electrophoresis electrospray ionization chip here. So the separation as well as the ionization happens on the chip. The sample is then or the analytes, the compounds, go into a mass spectrometer where we do identification and quantification, and then the Rebel does the data processing. So you don't actually see any peaks or anything, you just see the end result of the concentration of the amino acids in your cell culture media at the time when you analyzed it. So just a quick word on analysis for life by 908 Devices. Our company has, as I've mentioned, this Rebel system. Inside it, it has a high pressure mass spectrometer, which is also used in our handheld devices, which are used for looking into drug contamination or explosives, etc., by the army, by the police, by firemen, handheld device which is then just the mass spec itself. It doesn't have any separation. But then on the other hand, the Rebel also has inside these chips, which is the capillary electrophoresis. And these chips you can connect to a mass spectrometer by a zip chip device. And that is also something that our company, 908 Devices, then has in the product portfolio. In all of these cases, we want to note that the analytics and the machine learning is kind of inbuilt so that these devices are easy to use information at the point of need. So the Rebel that we talked about uses very little sample, so you can take 10 microliters, 30 microliters, around there is what you need to dilute and then shoot. And it is of course cheaper than a mass spectrometer you typically would see in a lab, and it is actionable at line device for you. Now, when it comes to life sciences, I always need to, when I represent 908 Devices, I have to mention the sister or the brother product, the zip chip, which then is for life science research, for example, for metabolomics, and of course characterization of proteins, and so forth. And that would be all from my side, and I would be very happy to answer any questions you might have.
Excellent. Thanks so much, Mila, for that presentation. I actually have a question that might be applicable for both of you. And it's sort of, how many technical replicates do you usually test or recommend, or is it just really one sample that goes in?
So from my side, the analysis only takes 10 minutes. I would always, as an analytical biochemist, I would always recommend taking 3. It kind of depends on what is the need. So the precision, precision and accuracy, if you're okay with just knowing that it hasn't depleted, if you're not wanting an exact number, you can get away with just a single analysis. But as an analytical biochemist, I can't really state that single would be enough. So I have to say triplicate.
Yeah, am I still, you still hear me?
Yep.
Yes, we can hear you.
I say yes. Yeah, I 100% agree with Mila. I mean, to me, replicates depends on the deviation within each group. So if you look at animals and humans, which have much larger variations, you want larger group size. So for something like cell cultures and media, typically the standard deviation is very low. But like Mila agrees, I really don't like doing one-offs because you just don't always know what may have happened on the way to the door. So I prefer replicates of 3, but certainly we've done one-offs and looking at certain time points as well for different, in this type of example, where people are giving us, just to cut down the number of samples. So if they're doing a large number of time points and different culture conditions, then multiplying everything by 3 gets to be pretty expensive. So we do things one-offs. And again, since you're doing a time course study, everything should show some type of, at least the abundant ones, a nice pattern. So there we do one-offs. But I like Bill, I prefer 3, but 1 can work in these cases, yeah.
Great, thank you. Mila, there's a question in the chat. Is the zip chip a MS source interface? Is it applicable to most MS systems, such as those from Thermo or Agilent, etc.?
So currently we are applicable to Thermo Scientific, to SciEx, and to Bruker as interface. So Agilent and Waters, to my understanding, are not included. And this is then for different reasons, but Thermo and SciEx and Bruker. And if you would like to learn more about the zip chip, please I have my email address somewhere. But also then you can look for some of the publications, etc., online. Just the 908 devices and SIPCHIP. You should find information there. I didn't focus on it today.
Thanks.
Yeah.
Awesome. Well, I know we're at the top of the hour, so I want to thank both of the speakers today for sharing all that knowledge and insight. I think this is a really important topic for this industry, and hopefully some companies and groups can reach out to you to take some further discussions offline. So thanks everyone for attending, and we'll see you back here next month.
Yeah, thank you everyone. Thank you for this opportunity.
Thank you. Bye-bye.